Welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Friends, today is Halloween, and that means we're back again with another scary pest. This time, we're looking at fall armyworm, an insect with an appetite that stretches across the globe, and more importantly, the dynamic duo of Sandra Woolfolk and Leigh Hawkins, who are teaming up to halt its destructive march. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Sandra and Leigh with us. Sandra Woolfolk is an Indonesian native who earned a bachelor's in agriculture from Gadjah Mada University in Indonesia. After she received her bachelor's, she worked for about 6 years before going to graduate school in the U.S. She obtained a master's in entomology with specialty in insect microbiology and a PhD in life sciences with specialty in entomology and plant pathology, both from Mississippi State University. She's worked in academia and in the private sector. She's currently employed with USDA-ARS's Corn Host Plant Resistance Research Unit as a research entomologist and has been in that position for almost 3 years. Throughout her entire career, from undergraduate degree until now, her job has always involved raising insects. Leigh K. Hawkins earned a bachelor's in biology from Tougaloo College in Jackson, Mississippi. She then completed a master's in horticulture at Mississippi State University and a PhD in horticulture at Auburn University. She did postdoctoral research with the USDA at North Carolina State University and has been employed with the USDA-ARS Corn Host Plant Resistance Research Unit as a molecular biologist since 2001. She spent most of her academic career avoiding insects, but here she is now with the bugs. She truly enjoys the variety of fieldwork and lab life. Hi, Sandra and Leigh. How are you both doing today?
We're doing well. How are you?
Good morning.
Good morning. Yeah, I'm so excited to record with you today. So it's getting to be Halloween times, and so that is a time of the year where we like to celebrate the work that researchers are doing to fight a scary pest. So today's pest is fall armyworm. So to get us started, I'm hoping you can just give us kind of an overview of fall armyworm, what it is, what it does, what it looks like, where it is. Just tell me everything about this scary pest.
Well, for that, probably I will talk about it a little bit since I'm the bug person. So, fall armyworm is actually native to Americas, and it is a global insect pest. And I would like to emphasize the word global because that's what makes this insect scary. This insect is invasive to other continents, is capable of long-distance dispersal. And also, the caterpillar life stage is the stage that damages the wide variety of crops and causes large economic losses. It is highly polyphagous, meaning it has been reported to attack more than 350 plants from 76 plant families, including corn. Now, in corn, these caterpillars injure leaves, ears, and tassel tissues. It was estimated that the annual value of corn yield lost to fall armyworm was around $300 million US, and now it's rising to about 500 million US or more in major outbreak years. There are many efforts have been done by several groups to manage fall armyworms. You want to talk about what we do as our corn unit for managing that?
Yeah, please.
Since our unit, USDA-ARS unit, Corn Host Plant Resistant Research missions is one of them is to identify and develop corn germplasm with genetic resistance to insect damage. In this case, it's fall armyworm using conventional breeding methods. So our effort is ultimately to identify genes or pathways associated with fall armyworm resistance in corn lines using a variety of tools and analysis. Now, two tools that we chose recently were called GWAS. GWAS stands for Genome Wide Association Study, and PASS. PASS is Pathway Association Study Tool. Both of these tools allowed us to identify regions within the genome or genes associated with resistance. And then PASS help in identifying metabolic pathways that may provide new resistance mechanism. Based on the outcome of our research, these two tools we hope to be able to continue and contribute to making of fall armyworm resistant corn cultivars. I hope that helps.
Yeah, it really helps. That's a great overview and leads really well into kind of the next section, which is just defining some acronyms that might come up. But before we get there, I wanted to ask, so you mentioned it's called fall armyworm, but you mentioned a caterpillar stage. So does this turn into like a moth or a butterfly, or is it just kind of worm-like its whole life? Like, what does this thing look like?
So this is moth. So the entire life cycle start with egg when they lay on the plants, on leaves or whatever plants. But for us in corn, they lay eggs in part of the corns and then turn into caterpillar, or a more scientific term is larvae. And from there and turn into pupae, which is like a cocoon stage, and then go into moth. So this is moth, so they're active at night. So hope that helps explaining.
It does.
Yeah.
It does. Yeah. And does it stay on the corn like its whole life? Is that where it makes the little cocoon, or?
Yes.
Does it move around?
Well, they move around, of course, at the moth stage. And actually, the caterpillar stage is actually, they're pretty active. They can move from one plant to another. So like in corn, if they ran out of source, they will move to the next plant. That's why I mentioned earlier, they're capable of dispersal long distance. They're very active even in the caterpillar stage.
Wow. Yeah, I was amazed to find out that they could go after that many different kinds of plants. They are hungry little critters, hungry caterpillars for sure. So, like I mentioned, there's a few acronyms that I just want to kind of go over in case they come up, or specified terms in this. So we talked about the Genome-Wide Association Study, or GWAS, which from my understanding is kind of comparing phenotypic traits, or the ones that you can kind of sense outside of like looking at the DNA, comparing that to the actual DNA genomic information to kind of match the two. And then the past you kind of touched on here, and we can go in a little bit more. But a few more that I wanted to just cover lightly is GBS, SNPs, and then can you tell us more also about these metabolic pathways and metabolites and kind of how that works?
Oh, sure. I'll take that question. So when it comes to GBS, that was what we call genotype by sequencing. And so we don't know a lot about the sequence sometimes, so we're able to get little snippets of the DNA and sequence it to see where there are differences. And we catalog that information. And so that gives us a type of markers called SNPs, which are single nucleotide polymorphisms, because we just know we just have a mutation at that one point. And then when it comes to GWAS, we take the information that we got from the GBS, which gives us a catalog of all the SNPs, and then we can compare that with the phenotypic data. And then it's called genotype, my words are stumbling now, genome-wide association study, because we look at the whole genome to associate the trait with the SNPs.
Okay. Yeah, and you guys in your worksheet had a kind of like a highway analogy, so maybe you could go over that to just solidify it in my brain.
Well, that's the easiest way to describe it, because if you think of DNA as a highway, just this long strand of just miles and miles of DNA. And along the way, we have these signposts. You have all kinds of markers on the highway. And those would be in this case, our SNPs. They're just a different type of DNA marker. And at some point, you have something that you're attracted to at some exit. There's several different things that make you want to go down this pathway. And so we assume that once we get to our the thing we're interested in seeing, that would be that attraction that are our trait of interest. And so we want to explore what's around in that area. And so the GWAS helps us identify all of those markers that are associated with our trait. And once we get off on the highway, say we're looking for a hotel, but when you take the exit, there's restaurants and there's all kinds of gas stations and why they're associated with the hotel, because it's there for the trait of interest, which is to provide comfort or whatever it is. So we just know that GWAS helps us pick out the big markers, and PASS, which is pathway association mapping, it helps us look at the other things that are not quite as significant but are certainly associated with our trait. I hope that makes sense.
That does. Yeah. That's super helpful. And then can you talk about the metabolic pathways and metabolites and what those are?
Oh, sure. We have all types of metabolic pathways that we experience all the time. A great one is photosynthesis. When you start off with some raw materials or your substrate, you just start off with like sunlight and carbohydrates and water. And the plant is able to transform that into energy. So that's what happens with our metabolic pathways. You start with a substrate, and there's several sequences of reactions that occur that lead us to our ultimate goal. So this is just one example of a metabolic pathway.
Okay, so the pathway is kind of the process by which something is created or accomplished.
Exactly.
Perfect. Okay. All right. Thank you for laying groundwork with me. Genetics papers, anything genetics papers can be a little complicated, so I always like to make sure I fully understand before we proceed. So, as you lightly mentioned before, we are talking about kind of finding some pathways or mechanisms for resistance within different corn lines to resist fall armyworm. So you had kind of two halves of this project, including some field work and some lab work. So I'm hoping you can tell me about both of those.
I'll take the field work. So we have this numerous diverse corn lines that we planted in the field. So we selected 289 to be exact, that were resistant and also susceptible to fall armyworm. We already know that at the beginning. We did the evaluation in the field or trial for 3 years. And it's located on campus at Mississippi State campus because that's where we're located. And we follow standard corn production practices when we did this study. And for our study, that's the field part, but so there are two parts, even just the field part. We also have lab. My lab is actually raising insects. We raise this annually, and one of the insects that we raise is fall armyworm. We raise a bunch of caterpillars before the study. And the first stage of the caterpillar, normally we call it neonates. We use it for this study. And we brought it and infested into the world stage of corn, or also known as V7 or corn seedling at 7-leaf stage, to be exact. And we use a special applicator and we put them in the world stage. And then after 7 days later, then we do the rating. Also 14 days later, we rate the damage. We're not just randomly rating. This rating has been published before, so it's a standard already. Now, after that, we take the data, we statistically analyze, and then at the same time, we also collect the samples, the leaf samples, and we brought it to the lab where Leigh started doing all this analysis, molecular biology analysis. Hope that makes sense.
Hi everyone, I hope you're enjoying the show. Interested in learning more? Sandra and Leigh's paper, Genes and Genetic Mechanisms Contributing to Fall Armyworm Resistance in Maize, published in the Plant Genome, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Let's get back to the show. It does. I have a question about this. So you mentioned some of it is happening in like a greenhouse setting with the infecting them at the whorl stage with the 7 leaves, and then taking the 7 and 14-day data. For the field data, are there like precautions that have to be taken to make sure that they don't just turn into moths and leave and go infest more stuff? How do you, is it in a greenhouse setting? Like, how do you manage that so that you're not accidentally just letting them out into the world? I mean, I assume that's something you're actively trying to avoid with a mobile pest like this.
Actually, this is in the field. It's open field. It's not in greenhouse.
Okay.
Yeah, so it's 7 days, because just keep in mind, this is global pest, and then second, what we planted, most of these we have susceptible plant, we have resistant. Some of the resistant, they probably won't even survive after our evaluation because of something within the corn that they don't like, so they just don't survive. So we plant it in an open field. So 7 days we collected them. We do the rating, and then what I'm trying to say, it's they probably die for the resistant one. And also surrounding, we have I didn't explain here. So each experimental plot, we have what we call it fillers. Fillers mean corn plant that is very resistant, like BT corn plants. So it's surrounded with these plants. So anything that escape will go to that plants, and they will not survive because it will die with, they will try to eat, but it will just kill them. That right, Leigh?
Oh, okay.
Does that explain well?
Yeah. Yeah, I love that, because it's almost like, it's like a fence almost.
Yes, it's like a fence. That's a good analogy. So we fence it with the very, very resistant corn plants that will kill fall armyworm.
Oh, I love that. How big are these plots?
Oh, it's pretty big. So we have 3 replication. So I rounded up, say 300 plants, 3 replications, so it's 900 plants total.
Okay.
So that's pretty big. This is one of the biggest experiments that we ran. So
fun. Yeah, I love that. I'd always like to have like a visual in my mind of, is this just like, 5 feet by 5 feet and there's just a ton of them? Is it one big thing? So that is helpful. So you mentioned already kind of gathering this data and doing some of the analysis on it. So I'm assuming that's a nice lead in to kind of the lab work that you were doing to do all this genetic testing and GWAS and PAST and all of that stuff. So tell me more about that side of the process.
Yeah, that's the part where I get to go to the field and start collecting the tissue. So we time the tissue to collect tissue that's in the world of the plant. And once you get into the world of the plant, as the leaf is maturing, it starts off with like a yellow-green color and eventually becomes greener. And so we collect some of that tissue. And from there, we go to the lab and we extract our DNA. And then we use the DNA we collect to associate with the ratings that Sandra gets from the field data. Because ultimately we know there's something that the armyworm does not like in the corn, and we want to find that. And so we use DNA to kind of give, in those markers we were talking about, the SNPs, we want to see what's associated, to see what could it possibly be, where could it possibly lead us. And we do a lot of analyses, there's a lot of statistics involved. So then you want to look to see, is there a pathway associated with any of these markers? And once we find that pathway, it's like, then that gets more interesting because we want to see what is on that pathway. Like, there's got to be something here, because something, the plant, the corn does, the armyworm doesn't like this part of the corn, so why does it not like it? And in this case, we came up with a couple of different pathways, but we decided to measure, I think, just two of them. We looked at one that measured carotenes, which are involved with photosynthesis. It's just a pigment in the corn. It's naturally occurring, and it gives carrots that orange color. So a lot of vegetables are orange because of carotene. And something that the armyworm doesn't like, something about that, what we don't know quite yet because we're still exploring that. And we also found something what we call cuticular waxes. And when you think about cuticular waxes, these are just, some plants have that waxy substance on the top of the leaf, and it kind of keeps it from, it helps control water evaporation. So something about that, the armyworm doesn't like also. So it's like, it's something about it. We haven't figured out what it is exactly yet, but we're interested in exploring those kinds of things.
Oh, I love that. I particularly like the wax because I'm just thinking of like when people accidentally like bite into wax fruit at like somebody's house and they're like, this is not what I thought it was. Exactly. And that's really pleasant imagery for me to imagine. And I'm also just imagining like carrot orange corn plants that are just resistant now, which is for sure not probably how that works. No, I had a, okay. So these mechanisms for resistance, you mentioned a couple. So are these kinds of things like, is it just like, I don't like this taste or this texture? Is it like, can they actively be poisonous? Is it a hormone thing? Or is it just like, when you're looking for these, is it like everything's on the table, we have no idea necessarily like what it is, and that's what the SNPs are for? Like what are maybe some of the categories of things you might be looking for, I guess is what I'm trying to ask.
I think the thing is we don't know what we're looking for. And this is why we use the SNPs. That's why we're searching the whole genome to find what could it possibly be. We know something is there. We just don't know what. And the SNPs will hopefully point us in the right direction so that we can explore more to see, is it hormonal? Is it something they just don't like to eat? Is it something they find toxic? We just don't know. And so this helps us it's more of an exploratory tool to lead us to a more direct answer. I
love that. Yeah. Oh, gosh. I'm so excited. I love that science is just like, there's always more questions. I'm a big fan. Mm-hmm. So you mentioned already a couple of SNPs and pathways that you were investigating. What else did you find?
We found there were some other genes that were interested or that looked interesting, but they're not always associated with a pathway, which means maybe there may be a pathway we don't know about yet. Just more things to question. And there's several other pathways that are of interest, but we just picked two metabolites that we can measure pretty quickly. But there were several other more. I think there were 31 pathways that were really of interest. So we have a lot of work to do.
Yes,
yeah, for sure.
Yeah, we can.
Were there any surprises in your research?
The biggest surprise to me was just seeing the difference between the genes and pathways that were significant at 7 days versus the ones at 14 days. Sometimes there was some of the same pathways, but sometimes they were slightly different. And so that's just a reminder that when it comes to genes and gene expression, it all matters about when it's produced, how it's produced, how much it's produced. All those things just vary so much. And there's so many things to keep track of as you're doing this kind of work.
For sure. Yeah, I am always impressed with researchers and with farmers who are handling just like the practical management of this day-to-day of just how many factors there are to consider. I am always impressed. So thank you very much for doing this research. And obviously this has some broad-reaching implications as we continue to fight the good fight against fall armyworm. So what are some big implications for this research?
Well, I can take that question. So we can this can provide additional means or avenues to study for fall armyworm resistance, particularly more targeted analysis of the pathways that were identified, and also further to do gene expression analysis. Like Leigh mentioned earlier, there are so many that we don't know, so we can explore more in that area. And also, like she said, about some pathways were associated with damage at 7 days, and then 14 days are something different. Well, we maybe can do study where timing is key and could lead to some more interesting such studies. There's a lot to explore. So, yeah.
For sure. For sure. And so that kind of covers some of the broad areas for your own future research. But in general, well, I guess if you have other aspects of your own research, but also kind of more broadly reaching, like, what are the areas of research moving forward as we continue to combat fall armyworm?
I can give some example for future research that I'm always looking at it from our perspective as a corn host plant research assistant unit. Leigh may be able to add some more to something that can be done, perhaps using markers or genes that were identified for gene expression studies, or interaction between genes and pathways with fall armyworm. I don't know, Leigh, do you have anything in general?
Those are exactly the kind of things we're interested in. We want to know more about these pathways. So they've kind of been identified. Now we want to know along the pathway, is there a certain trigger that leads to resistance, or something? Just what is it? We don't know at this point. So we're just trying to. It's just we have so much more that we can possibly do. And so those are the kind of things that we're always interested. We get some answers, but then, like you said, Abby, leads to more questions, and it keeps us engaged constantly. And those are the kind of things we want to focus on.
Yeah, for sure. Sounds like there is indeed very much research to still be done. So I have 3 questions left for you so you can get back to said research. The first one is, if listeners want to learn more about fall armyworm or your research or any of the things we've talked about today, where can they go?
There's actually a lot, numerous information online. Since again, this is global pest, so there are so many areas and places. Even Africa is one of the continents that hit hard by fall armyworm. So Wikipedia has information. I'm just giving example. You can just put in the web search Wikipedia, and Mississippi State Universities, where we're located, they have also the extension of the Mississippi State, and then FAO, all as well as USDA. You can just do web search. It's just unlimited. And of course, you can talk directly or pick up phone call to their extension office.
Sure. Yeah. And we'll have your contact information in our show notes. Obviously, you are both experts in this as well. Leigh, did you have any other resources to add on that question?
Well, I think Sandra did a great job covering those. I know we have a link to our research unit, and you can find that at arsusda.gov. And there's also some links. You can find USDA on LinkedIn and on Facebook, and sometimes you can find some more information about that. But the best resources would probably be using your extension agents or just Wikipedia, some things on the internet, because fall armyworm is literally everywhere.
Mm-hmm. Yeah, for sure. So then, if people want to take the next step and get involved in this research, and that could be as researchers, as farmers looking to improve their resistance on their farm, civilians, whatever. Those are also researchers, and farmers are also civilians, but general public, I should say. What can they do?
Well, I think one of the greatest things you do, if you can kind of look out in your yard to see if you even see fall armyworms. I think a couple of years ago we had a big outbreak of fall armyworm on Bermuda grass, and people were reporting it all over the place, and that helps us get more information about where the insect is spreading. And it just helps us with our research also, just if we want to look at the impact of what we were doing and what is happening.
Sure. And what do they look like? I forgot to ask.
Sandra can do that better. I do the research side. She does the bug part.
Well, they're kind of grayish, dark. Has a so overall color maybe more dark grayish. It's hard to describe it in words. But the head, you can kind of see a Y, inverted Y on the head capsule. And also at the posterior is the end, or like the tail of the caterpillar. I'm talking about the caterpillar. It has like 4 dots, like squarish, forming square, but it's in a form of 4 dots. If anyone want to go to internet, like Leigh said, Google fall armyworm. It can be Google or anything else, but just find image. You'll see it. You can tell. There's so much information out there.
Sure. Alrighty, so I will leave it to listeners to look that up and also look up the moth version. It's extra incentive. We're not going to tell you here.
The moth is it's kind of I'm not sure how to word it. It's more tannish, but it's hard to distinguish with other moths unless you really look at it under microscope, or if you're used to it, to look at it every day. So caterpillar is easier to tell.
For sure. Yeah. Tan moth sounds like every moth I've seen in my backyard this year. No, that's really great information. That's a great way to get involved, and kind of like a little citizen science vibe. So I like that too. So last question for you then today is, what is one fun fact that listeners wouldn't know about you if all they had was your research?
Well, if it's just one, I have several, but one fun is I like to adventures to try different kinds of food, different, from different countries, different ethnic. That to me is, adventure in the type of food. It's fun to try out. That's yeah.
What's your favorite?
I actually, besides, because I'm from Indonesia, of course, my favorite is Indonesian food. But beside that is Thai food. I always like spicy food and their flavor. So yeah, I enjoy it a lot.
Yeah, Thai food is great. Big fan.
Yes.
Leigh, what about you?
Well, I was saying that I would encourage people to go visit Natchez, Mississippi, which is where I grew up. And it's one of the oldest cities on the Mississippi River, and it's a very unique little place.
Ooh.
It's beautiful, and I always tell people to go in October when they have the balloon races over the river, but that's my plug for Natchez.
Ooh, nice. Well, this is launching on Halloween, so this is your last day this year. Or next year. Put it on your calendar now.
Yeah.
Well, this has been such a lovely conversation. I have loved learning all about the efforts that you are putting in to halt the progress of this Halloween scary pest. So thank you so much for the research that you are both doing and for taking the time today to talk with us. And I hope you have an awesome rest of your day.
Well, same to you, and thank you for having us.
Day.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.